All right, let's continue on with this non-Mendelian genetics. So the next one we're going to look at is epistasis. And epistasis is a situation in which one gene masks or otherwise affects the expression of another gene. So this is seen with Labrador retrievers. Now, Labrador retrievers you can either have a black lab or a chocolate lab or a yellow lab. To get a black lab, you have to have a dominant allele in both of the genes that are for coloration. All right? To get a chocolate lab, you have to have two recessive alleles in that first gene and a dominant allele in the second gene. All right? To be a yellow lab, you need to have two alleles in that second gene. All right? It doesn't matter what the first gene is, whether it's dominant or recessive. All you need to have there is two recessive alleles. All right? So this is where one gene affects the expression of the other gene. All right? So that's showing epistasis. Next, the one that this thing kept trying to switch to is what is known as incomplete penetrance. An incomplete penetrance is a genotype that does not always produce a phenotype. So an example of this is what you see with this little kitty cat here on its paw. You can see it with this next kitty cat here on its paw, and that is polydactylism. So these guys, even if they have the gene for polydactylism, they might not always express that gene. They also show another trait called variable expression. And this is where a phenotype varies in intensity in different individuals. All right, so here, you could have an extra digit on one paw, or you could have extra digits on both their front paws, or any other variations that can arise. All right, let's move on to pleiotropy. Pleiotropy is where one gene affects many phenotypic characteristics of an individual. And this is what we see with a lot of these single gene traits like cystic fibrosis and albinism. In albinism, you don't produce melanin, so that affects your hair color, your skin color, your eye color, right? This is showing sickle cell disease, and because you have this abnormal hemoglobin, this produces these abnormal red blood cells. And what happens with these cells is because they have this abnormal shape, they break up in our blood vessels much easier than typical red blood cells. So you see this breakdown of red blood cells, they can clump together, and then it can cause all of these other problems to arise. All right? So now let's look at incomplete dominance. An incomplete dominance is a situation in which neither allele is dominant. And that's what we see here. All right? So this is shown with snapdragons. Snapdragons are a type of flower, and so here showing a red snapdragon, and if you cross a red snapdragon with a white snapdragon, all their offspring in F1 are going to be pink. And so what we see with incomplete dominance is that this is a trait where if you have more of the genes for a particular trait, you show more of that trait. So here, you can see how we do this a little differently than before. We don't use a capital letter and a lowercase letter. We use a capital letter, and then we use that trait in the superscript. All right? So here, this is the color, right? And so, since this is red, we call it the red color. So this individual has two alleles for red color. This individual has two alleles for the white color. But white really is a lack of pigmentation. So this is red pigmentation. Here's a gene for no pigmentation. So when they have offspring, those offspring are going to have half the pigmentation of those that have that red coloration. And half of that pigmentation is pink. Okay? So two genes for pigmentation, a nice dark red. Two genes for no pigmentation, white. One gene for pigmentation, one gene for no pigmentation, you get this pink coloration there. So if you cross individuals who are heterozygous with each other, you'll see this 1 to 2 to 1 ratio play out. So what you see here, right, is that the phenotypic ratio, 1 red to 2 pink to 1 white, is the same as a genotypic ratio, which is 1 CRCR to 2 CRCW to 1 CWCW. All right. Oh, this is showing this with cholesterol. So cholesterol is also an incomplete dominance inheritance here. So if you have two genes for making these cholesterol receptors, known as LDL receptors, you're going to have genetically low cholesterol levels. If you only produce one gene for this, you produce half of these LDL receptors that an individual would produce two. So you're going to have a mild disease. Your cholesterol levels are going to be a little higher, all right? And this individual doesn't have any genes for these LDL receptors, so they're going to have genetically high cholesterol levels because of that. All right, let's look at polygenic inheritance. So poly means many, genes, polygenic, inheritance. So this is a trait that is controlled by several genes. Most of the traits that you're going to associate with a person are polygenic. So their eye color, their hair color, their skin color, their height, weight, these are all polygenic traits. Many genes are going to determine the trait. All right? And once again, just like incomplete dominance, they're going to have an additive effect to them. All right? This is showing an example of two individuals, up here at the top. And this is showing the skin color. So if you have individuals that have three alleles for darker pigmentation and three alleles for light pigmentation, and those two individuals mate or have children, they could have children of any shade of pigmentation in their skin. Some that are very light to some that are very dark. All right. So, as I said, when you look at individuals, the thing that we see most, all of these are polygenic traits on these differences between these individuals. Now, let's take a look at the environment, because both the environment and the genotype are going to affect the phenotype. The importance of this can vary, right? So, for a lot of us, we go outside in the sun. That sun exposure is going to cause our melanocytes to produce melanin, and we're going to get a tan, all right? So that's a slight change in our phenotype, all right? Nutrition can also affect your height. So the nutrition that you get while you're growing can also determine how tall you're going to be, right? So what we found out is that your height is actually 80% your genes and 20% the environment, mainly your nutrition. And this is why we see that people from industrialized countries are typically taller than people from non-industrialized countries. And that's because we typically have a better diet, all right? So that's a little bit more importance on the environment than what we saw with, skin color change. All right, here's one that's even bigger. So there are some organisms that do not have sex chromosomes, like turtles and crocodiles. And so with them, it's the temperature of incubation of the eggs that determines the sex of the offspring. So with turtles, if the the nest where they buried the eggs is a little warmer, they're going to have more females. If it's a little cooler, they'll have more males. With crocodiles, it's the reverse of that. So that's a big influence of the environment on the genotype, on the phenotype. Here's showing a couple examples. This Siamese cat and this Himalayan hare both share the same trait, where if their skin's temperature gets below a certain degree, they start producing darker fur in that area. So this makes sense for the Himalayan hares. They're found in the Himalayan mountains, which is a mountainous area, so they're going to have a lot of snow. So they want to be white to blend into the environment. So, but if their temperature in their skin gets below a certain temperature, a certain degree, they will then start producing dark fur in that area because it's going to absorb more sunlight and make that area warmer. This is showing hydrangeas. Hydrangeas, it depends on the pH of the soil. So here, pink hydrangeas are grown in a basic soil. Blue hydrangeas are are grown in an acidic soil. So you can add vinegar to the ground to do this. You can add lye to the ground to get it to do this. Here's the last example of environment. So these are both red tulips. So these are red tulips, and then also these are red tulips. But these tulips are infected with a virus that causes this yellow streaking to occur. And this is at my old house. I had red tulips there. And my wife used to love these kind of tulips here. She would say, oh, I like those striped ones. They look so pretty. And I would say, yeah, they're infected with the virus.